Hello, and welcome to the Fluxergy training module on how to run a PCR test with incubated samples. Today, we will be discussing the materials needed, each step of the test kit process, and important notes for using our analyzer for Fluxergy PCR test kits with incubated samples. The general overview of running a Fluxergy PCR test are the following steps: sample collection and incubation, test setup, loading the test card, running the PCR reaction on the Fluxergy analyzer. For running a PCR test, you will need additional accessories outside of the Fluxergy portfolio. We will now go over the materials required for using our test kit. For exact specifications, please see our instructions for use for your test kit. For sample collection and enrichment materials, you will need an incubator to grow any present specimen at a detectable amount. You will also need a mini vortex to homogenize your sample, and you will need pipettes and filtered pipette tips at 1,000 microliter volume. For sample reaction mixture preparation materials, you will need a 20 microliter and a 200 microliter pipette and tips. We recommend using dedicated calibrated pipettes and sterile barrier filter pipette tips to avoid contamination between handling samples. You will also need a mini centrifuge to spin down your reagents and ensure a complete volume can be collected from the vials. Now let's get into performing sample collection. Review the instructions for use for your PCR test to determine which validated sample type to collect. The environment is ready for swab collection once it has been cleaned and dried. Your Fluxergy Salmonella test kit will come with one of two types of environmental sample sponges. These sponges will provide the same quality results with only minor workflow differences. Please follow this set of instructions for sample collection for the sponge stick swab. Hold the sponge in place inside the bag. Shake the stick to the end of the bag and tear open the bag. Aseptically, grasp the sponge stick from the handle and remove from the bag. Now swab across the sampling surface according to your organization's swabbing protocol. Swab on both sides of the sponge. Once done swabbing, place only the sponge component of the swab back into the bag and bend the stick back and forth at the base of the sponge to break off. Allow the sponge to drop into the bag and discard the stick handle. Roll the bag to close and fold ends of the blue wire inward to secure closure. If you have a sponge swab with no stick, please follow this set of instructions for sample collection. Shake the sponge to the end of the bag. Tear the bag open and push the sponge so that it extends from the bag. Bend the blue wires to form an open bag. Then, set it down on a hard surface. Put on sterile gloves and remove the sponge from the bag. Swab across the entire sampling surface. Then, turn the sponge over and swab the same surface area again. Place the sponge back into the bag. Roll the bag closed and fold the ends of the blue wires inward to close. Once collected, the sample should be incubated in the Fluxergy-provided selenite broth for at least 16 hours. The incubation process should occur within 48 hours of sample collection. However, performance is not guaranteed if samples are not incubated immediately. To incubate your sample, first, obtain the 20 milliliter vial of selenite cysteine broth and uncap. Obtain your environmental collection bag. Move the sponge to the top of the bag and fold the bag in half. Squeeze to mix contents of the bag to ensure homogenization of specimen and neutralizing buffer. Open the bag and using a 1,000 microliter pipette, draw 1,000 microliter of the sample. Do not allow the pipette to touch the insides of the bag to avoid cross-contamination. Dispense 1,000 microliter of the sample into the selenite cysteine broth. Do not allow your pipette to touch the inside of the vial to avoid contamination. Eject your used pipette tip into a biohazard bin and cap your selenite cysteine vial. Make sure to label your vial with the sample name and date. Vortex and invert vial 10 times to mix contents. Then, slightly loosen the cap to ensure proper aeration of the media. Place the vial into an incubator set to 37 Celsius for at least 16 hours. Do not enrich the sample for more than 24 hours. If not testing immediately after incubation, please refer to the selenite cysteine broth's instructions for storage. You are now ready to perform a PCR test after sample enrichment. After 16 to 24 hours, remove the incubated sample vial from the incubator and close the cap thoroughly. Then, take out a Fluxergy reaction mixture vial from your negative 20 degrees Celsius freezer. Each vial is single-use and is viable for up to 30 minutes after thawing. Please only thaw for the number of tests that you'll be performing immediately. To thaw the reaction mixture, you can do so by quickly holding the vial in your gloved hands for approximately a minute, or leaving out in room temperature for 4 to 5 minutes. After defrosting, bring your reaction mixture to your test performance station with your other materials, including your incubated sample, pipette and tips, mini centrifuge, and test card. You are now ready to load your test card. Take out the test card and its cap from the packaging pouch. Keep the pouch aside so that you may register this test card into our software. Vortex the sample or invert 10 times to homogenize the contents. Grab your 20 microliter pipette and attach an appropriate sized barrier tip. Ensure that your pipette is set to the indicated volume per your test kit's instructions for use. Collect the indicated volume of your incubated sample, making sure to pipette slowly and that there are no bubbles in your tip. Do not toss away your incubated sample, just in case you may need retest. Next, dispense the full volume of the pipette tip into your defrosted reaction mixture and flick 5 times to mix. Then, place the vial in a mini centrifuge so that the full volume of sample reaction mixture is not lost from adherence to the wall or cap of the vial. Then, grab your 200 microliter pipette and set the volume as indicated by your test kit's instructions for use. Again, ensure that you are collecting the full indicated volume from the reaction mixture vial. You should be collecting with the tip near the bottom of the vial, underneath the surface of the reagent. No bubbles should be present in the pipette tip. Now obtain your test card and locate the loading port. Eject the reagent directly onto the bottom of the test card loading port. We do not advise pipetting on the sides of the loading port, as you may lose reagents that cling to the wall. Toss away your reaction mixture vial in a biohazard bin. And lastly, cap your test card. You are now ready to load your test card into the analyzer and start your PCR reaction. When bringing the loaded test card to the analyzer, be careful to keep the card 180 degrees flat. While there is reagent in the test card, do not invert, drop, or shake the test card. Doing so will impact the volume of sample and reagent in the loading port that is available for analysis. Next, simply insert the loaded test card into the analyzer. Now we can set up your Fluxergy analyzer for testing. Obtain the test card packaging for your test card. Then, log into Fluxergy Works and click Run Test. You will be prompted to scan the barcode located on your test card's packaging pouch to initiate a test. If you do not have a 2D barcode scanner, select the manual entry option and follow directions listed in the dialog prompt. When you have entered your test card's information into Fluxergy Works, you may now record the remaining test information sections. First is the test name, which must be unique for each test. For example, you can enter the assay type, date, and run number. Make sure you select the correct test type and template corresponding to your assay. Finally, enter your sample ID as defined by your organization's protocol. You also have the option to include any other information not previously listed in the notes section. For example, you may want to note the brand of viral transport media or the sample type. Finally, click Run Test and select an available device. Your device is now ready to run a PCR reaction on your test card. Congrats! You are now ready to run your PCR test with our Fluxergy analyzer. Please clean your workstation with an appropriate disinfectant after each test to avoid cross-contamination and to prevent incorrect test results. For additional support, you may also complete a customer support ticket for questions or additional one-on-one training. Happy testing!